this should be recording now. I'll post it in my courses probably right at the end of lab. Great. Can you all see my screen? Okay. So brief introduction. Okay, first, yeah, if you need anything, use the chat. That'll be easy, or unmic and interrupt me. Raised hands probably aren't going to work because I can't see everyone at the same time. So yeah, just interrupt me if you have a question or use the chat. So basically today we're going to cover the syllabus, talk about the course a little bit, what's expected, how to submit assignments, things like that. We're going to cover the worksheet and hopefully give you everything you need to do your homework for this week. Didn't change that. So my name is Hunter Trowbridge. I did borrow this from Amanda, but this contact information is correct. So if you need to get a hold of me, that's my email. I sent you all a few emails now, so you should be able to get it that way. I'm going to have office hours on Mondays from 10 to 11, Wednesdays 1 to 3, and Thursdays 10 to 11. If none of that is going to work, hopefully that's spread out enough that it will work for everyone. But if not, email me and I'll set up an appointment, do my best to accommodate everyone. And I'll have a Zoom classroom set up for that on my courses as well. So learning objectives for this course, all of this is available in your syllabus. Just very briefly as an overview, a lot of what we're going to cover, designing experiments, using statistics, working on hypotheses, things like that, it's all really important if you're going to be a biologist or an ecologist or any related fields, pretty much, through the rest of your degree, and then immediately afterwards, these are going to be really applicable skills, things that you're going to need to build on. Hopefully we're going to have a good overview of that in this course. We use software, a few different programs in this course, which is a pretty common thing. Most statistics is moving towards R right now, which you may have heard of. We're going to use a program called JASP, but this is going to be a really good introduction for being able to manipulate data in statistical programs. Being familiar with that process will help you a lot in the future. Quickly, assignments and grading, there's total 900 points in the course. Attendance and participation is worth 25 points. I think I'll do attendance at the end of each class. Basically, you're going to have a worksheet that you need to do for each lab. So check in with me at the end of class, show your work, and that will count as your attendance for the lab for that day. Semester schedule, I would suggest printing this out and having it, or have it in an easily accessible location. Attendance is going to be required for all labs, even though this is an online course and it's going to be recorded. You have to be here from 2 to 5. If there is an emergency, let me know as soon as possible, and we'll do our best to accommodate you. But for the most part, attendance is just going to be required. You'll be able to submit all your assignments through my courses. 5 points a day are lost for late assignments. Worksheets will be due at the end of each lab day, so midnight tonight, the first assignment will be due. There were some technical issues with the other labs and that it was extended. So, we'll be flexible. Everything is a little funky right now with people's internet and things like that. So if you have an issue, just reach out, communicate about that and let me know and we'll do our best. We're not going to be unreasonable about tech issues or things like that. All assignments must be submitted in specific formats, so either a Word doc or a PDF. They won't be accepted as any other format, just because we probably can't even open them. So just please make sure that your assignments are in the correct formats. Academic honesty is a really important topic to cover. All of the work you do in this course should be your own work. UMass Dartmouth has really strict guidelines for this. All of your assignments are going to be run through SafeAssign, which checks previous lab reports from other students in previous years and even in different lab sections of this year. So using other people's work isn't going to work out in the first place. It's also not what you're here for. Please just do your own work. It's going to make everyone's life a lot easier. It'll make you get more out of the class, even if it's hard and you need to ask questions or come to office hours or things like that. It's going to be worth it to put the work in and have it be your own work. So everyone should have access to my courses at this point. And there should have been instructions and links to download all of these things. For right now, we're not going to worry about BioGraphR. That wasn't working for us. And Simutext will be something we cover in the future. For today's lab, we really just need Excel and JASP. I'm going to stop sharing. So before we move on, was everyone able to download Excel on their computer? I can't see everyone, so if everyone could unmic or chat or something. And great. Okay, it seems like everyone was able to. This will not work without that. What about JASP? Did that work for everyone as well? I guess you can just chat if it didn't work. That might be easier. Great. Okay, well, hopefully you don't have any tech issues with that moving forward. And so you should have had all the assignment materials. I emailed those to you as well, but you should have been able to find those on my courses. So some of this might be review for a few of you, but it's just really handy to have more hands-on experience anyway. So we're going to do some work with an Excel spreadsheet, just manipulating data, creating charts, and doing or using some formulas in Excel. Yep, and then transporting that into JASP and manipulating that data in another statistical software. Let's So if everyone wants to open their spreadsheet and maybe the worksheet now, we can start working through some of this. Does anyone have any questions up front? Great. So this is the worksheet that you all should have been provided. If you downloaded this before Wednesday, you may be missing some stuff. So you may just want to go back and download the one that I emailed, or the correct file is on my courses now. So quick point on organizing data. There are standards for how things are done. Typically, you want to have variables across the top, so different things that you're measuring. And then you're going to want to have your data points going down the side. And so all of this data set is associated with a study that was done on campus, LOMO study, which I will share. So there was an initiative started a few years ago to reduce the amount of mowing on campus. I guess UMass Dartmouth has 705 acres total of land, and about 200 acres of that are mowed, which is a lot of labor. It's a lot of use of poorly regulated small motors that produce a lot of emissions. And it's also a lot of possible habitat being lost to mowed areas. So some faculty put together a proposal to reduce the amount of mowing to 1 to 3 times per year. And they selected a few areas on berms, drainage ditches, and between parking lots. So just a few selective areas that people probably wouldn't use otherwise, just as a test space so that you're not taking over, a popular grassy spot. And the reasoning behind this, there's a few different reasons of why you would want to do this. You're going to enhance biodiversity and ecosystem services, so better habitat. You're going to have more organisms using that area, which is really important to consider pretty much everywhere now. There's a lot of published data on habitat loss and effects on wild populations and habitat fragmentation too. So if you have an opportunity to improve your habitat where you live or work, it can be a really big benefit to wild communities around you. I touched on time and carbon emissions associated with mowing a little bit. Campus aesthetic, is one of those things where some people may like the look of manicured lawns. There's definitely a lot of push in biological communities to sort of change that perception. Basically, the idea being, meadows are beautiful and not necessarily associated with poor upkeep or things like that. So, and a university is a great place to show this, to have test meadows and show people that they can be beautiful habitat and that you don't necessarily need a manicured lawn. So study design, they had paired plots spread out across campus. There were 5 pairs at 3 locations. 2, 3. Plots were 15 meters by 20 meters. And it was established in spring 2018, so it's becoming a larger data set. There's audio associated with this. I'm trying to not have that play. So this is just a nice graphical representation of study design. You would have a unmowed and a mowed plot, and they gathered data at all of these sites for soil moisture, organic matter, plant diversity, plant composition, and biomass. They also did sweep nets and pitfall traps to look for insect diversity. A lot of this is measured basically by going out and doing soil cores. And we can talk a little more about how some of that data is processed when we look at the data sheet. This would be, parts of this are going to be irrelevant now because we're not actually on campus. But basically, we're going to go and look at percent soil moisture and percent organic matter. And we're going to do just some basic statistical. So this is a few examples of differences in in what happens when you have a frequently mowed and an infrequently mowed plot. So you can see, increase in plant species, increase in insect species and sweep nets, things like that. We'll look a little more at some of this and how to make these plots in a minute. And then we touched on biodiversity and habitat. And so within a year, they had things like white-tailed deer showing up in these places and using them as actual habitat, which is sort of a nice anecdotal evidence of that the slow-mo theory is working for wildlife. Okay, so going back to data sheet. And your worksheet. So touched on this a little bit, rules of good data management in terms of columns and rows. Does anyone remember what I said or have any input on why data is set up like this? So you want to have your variables on the top, and you're going to want to have your data points in the columns. So this is going to be, it might seem nitpicky now to have to set things up this way, but it will make a lot of sense in the future, especially when you start using formulas or programs like R, things like that. It's also just established convention. So this is the way you're going to see data presented in journal articles, things like that. So it makes sense to get used to working with data in this way to start. Sorry, I'm just switching screens again. Does anyone know what metadata is? Anyone have an idea? Great. So Dylan said data that describes other data, and that's exactly correct. It's data about your data. So if we look in the Excel sheet, just move this. So you can see a metadata tab now. Basically, this is just going to go into, in here you've got mowing, plant species, all of this, dry soil weight. This is going to tell you what all of that means. And that's important because, if you leave this data set and someone tries to use it in a few years, they may have no clue what your abbreviations were or what your metrics were or any of that. It's also important to talk about types of data. So you can have nominal, discrete, continuous, or ordinal data. Nominal is a name, so name of the plot. Discrete is going to be only certain values, so counts of plant species in this case. Continuous data is going to be infinite, so something like dry soil weight could be, any number possible depending on the sample size you took. And then ordinal is categories. So grass cover category, frequently you'd see this done percent cover or something like that. All right. So now we need to measure soil organic matter in Excel. So you're going to want to have this file open. And has anyone used formulas before? Is anyone familiar with this? Okay, so basically, you can see up here when I select this cell, it shows you instead of, that was wrong. Instead of this number, it's going to show you this formula up here. So what this refers to is the different cells in the worksheet. So we've got K2 minus L2 up here, and that's just referring to this K2 minus this L2. And the reason we do that is because you've got wet soil weight, dry soil weight. So subtracting those is going to give you the amount of water in the soil sample, dividing by K2, which was the total sample, multiplying by 100, will give you the percentage soil moisture. You have to start these with an equal sign in Excel. So, and when you have it selected, don't click out of the cell because it changes everything. So you can also, here we can redo this. So you have to start with an equal symbol. Instead of typing it in, you can just select the cell. So again, we got K3 here minus L3 divided by K3 again times 100. Does anyone know why this didn't work? Why I have a negative number? So Excel is sensitive to order of operations. So without parentheses, it's not going to work. So you have to include parentheses in your formulas when you use them. So why doesn't everyone take a minute now and in your Excel sheet, go ahead and work on these formulas, see if you get these same answers here, and then we'll move on. Give everyone 5 minutes to catch up with that. All right, it's about 5 minutes. Did anyone have any issues? Make sense? So the next thing your worksheet's going to ask you to do is to reduce the number of decimal places here, which you can do pretty easily in Excel just by reducing these. And it wants you to reduce them to 2 places. And then calculating percent soil organic matter. I'm not sure if you went ahead and did that as well. I believe the formula is in, or the basic formula of it is in your metadata table. But basically it's dry soil subsample minus subsample ash. So basically they took a soil core, they let it dry to release any of the water from the sample, and they took a subset sample of that soil core, took the dry weight and then burned it because organic matter will burn. So your subsample ash weight is going to be whatever was left. So basically anything inorganic that did not burn. And so the difference between the two should be your organic matter from the sample. Which is why if you find the difference between those two, divide it by the original weight, you get percent soil organic matter. So hopefully you all got the same answers here. Nice thing about Excel is that once you have this, you can select, once you have your formula here, you can select your data and drag it down. And that's going to fill in all of the data for the rest of the set. So it's going to take this, you can see K3L3, K4L4, K5L5. It's going to move it down the set and apply it to the next parts of your data set. You're going to do that for percent soil organic matter as well. And so now the sheet's going to ask you to make a box plot for percent soil organic matter. So if you click the P header, it's going to chat. Hold on. Great. So someone asked, how do you extend the formula? So I'm just going to delete this. So you can select your cell and then go to the lower right-hand corner. Cursor is going to turn into a black plus sign, and you're just going to drag it down through the end of your data set like that. So now we need to make a box plot, which is going to be simple. If I can actually see my screen, Zoom is taking over everything. So you're going to highlight your whole column here. You can do that pretty easily by clicking P, or you can drag and highlight that way. Going up in your top toolbar here, click on Insert, and then statistical charts. So you're going to go to Insert, Statistic Charts. It's going to give you the option of histogram or box plot, or box and whisker as they call it. Histograms in Excel are a little messy. You can make them work. We'll talk about that more. I believe Tara is working on a video for how to format things in Excel the way she wants them. But for now, we'll just cover box and whisker because it's a little bit easier. So that's going to bring this chart up in Excel, and doesn't look great immediately, but you can open with this little plus sign your chart elements. You're going to want to get rid of grid lines, get rid of your chart title. You want an axis title, and you don't want a legend. So basically you just want axis and axis title. And then the other important thing is that you either want your chart in grayscale or just clear like this. So you want no color. Changing the style, added everything back that we don't want. So if you, now Excel is putting this out with blue outlines, which we don't want. So if you click on it and right click, you should be able to change your outline color. Change the wrong outline color. There, now it's black. So that was trying to change the outline of the whole thing. Might take a few tries. You just have to get it to select the actual box plot like that, and then change the color. So two tricky things we're going to cover is copying and pasting out of Excel. So you can right click on your chart, copy it, move into a Word document, and paste it. Can everyone see the Word document? Is it switching screens?
No, they can't see. They can only see your Excel document.
Okay, so I'm going to switch to, thank you. All right, now you should be able to see the Word document. So go in there, Control V or right click paste. And when you do that, it's going to give you paste options. And you always want to paste as a picture. So it is sometimes easier to go up here in the toolbar. This option is paste as picture. And basically, because everything is in the Microsoft suite, if you just paste it straight as a chart, it's still linked to the data from the table. So if you go back and change things, then it's going to change the chart in the future, even if you don't recopy and paste that in. So to avoid any of that, you always want to paste as a picture. Similarly, in Excel, you can run into that issue with formulas. So as you can see, when I click on each of these cells, up here it's showing you the data for those cells. And so what it's showing me is that this cell doesn't actually say 33.77. It says a formula. And so to get rid of that, pretty much the same thing, except instead of paste picture, you want to paste number, paste values. And so what we just did was copy all of this, paste it here, but now you can see that instead of saying this formula, it just says the number. And so that's just a good trick to know. You may run into issues where things aren't working, and that's a good place to start your troubleshooting. I'm going to delete that because we don't need it. And that should pretty much cover everything we need to do in Excel for today. Does anyone want a few minutes to work on that and catch up, or can we move on to JASP? Great. So we're going to take another 5 minutes. Unmute yourself, ask questions. Let me know if I can help with anything. I want everyone to make sure they got the step-by-step sheet that Amanda created. I sent that out probably 15 minutes before class. And that's just got really helpful information for, it may go a little more in depth into some of this. And if you get stuck, it should have some step-by-step help to go through all of this as well. Stephanie, so you said your numbers aren't adding up for soil moisture or for organic matter?
For
both?
the organic matter, the soil moisture is fine.
Okay. Did you include the parentheses in the formula?
Yes. Hold on. Let me see. Yeah, so yeah, it's the I don't know. So I'm having a problem with that. It's not working. And then I can't find that button that makes it go down to 2 decimals, like after numbers 2 after.
Yeah. Well, let's start with the first part, just trying to get this to equal
Okay.
this, and then the other one is pretty simple. So you've got your chart showing the same values here for dry soil subsample and subsample ash.
Yes.
Okay. And then you've got M2, so you're doing the difference between these. So dry soil subsample minus subsample ash in parentheses divided by dry subsample.
Yeah. Oh, I was missing the second parentheses, I think. What's the number say on yours for that one?
11.39.
Oh yeah, that changed it.
Okay.
Okay. And then the how do you get the decimals down?
So in the top, sorry, it's cutting off the top of, Zoom is cutting off the top of my screen, so I might have brushed over that. It might be hidden if you don't have your screen maximized, but in this number tab here.
Oh, I see it. I just made the screen bigger.
Yeah, so when I shrunk mine down, it hit it on me. And you actually can select all of your data and do that at once. So you don't need to do that for individual.
Oh, I see. Okay. Wait, my numbers aren't the same. So the 11.04 is, but then after that, it's not.
Because when you changed this formula, it didn't alter the rest of them. So you're going to have to probably redrag. Oh, my numbers just changed. So I might have done something wrong.
Yeah, because that's what mine's at, the 12.27. Okay, so that's right?
Yeah, that should be right now.
Okay.
I must have had something.
That's all I needed. Thank you.
Yeah, no problem. Angela, are you all set now with the percent soil moisture formula? So if you want to select more than one column, I'm not sure. You would just use the control key. So you could select all of this, hit control, and then click the P drop down again. Does anyone have any other questions? Everyone feeling pretty caught up to this point? Great. Okay, so we'll move on to JASP then. Hold on one second. So you're going to want to save all of your work into an easily located spot on your computer, probably just a folder that you have for this lab, break it down into week 1, 2, 3, etc., and then save all of your work in there just to make it easy to find, especially since we're going to switch back and forth between Excel and JASP a few times. It'll just be easy to have everything saved in one spot and organized. So file, save as, this is all changed now. I like just using the browse function because it lets you go through your computer, and then you can go to wherever you're going to save your data and do it that way. Next thing you're going to want to do is, so that saved this spreadsheet as an Excel file, and we're going to need to save it as a comma delimited file because JASP won't open Excel files. So file, save as again, browse to your location, and I've got Mo data in here. I already have it saved as an Excel file. So then if you open save as type, there's a few different CSVs. I believe they all work the same for our purposes. I've just been using this CSV comma delimited plain one. When you do that, you're going to get this pop-up window, or most of you will. It's just saying that it can only save one sheet at a time. And since we have multiple tabs on this Excel file, it's only going to save the open one as a CSV. It's fine. You can hit OK. And now you should have that saved as a CSV. Everyone able to find that? Anyone have any questions? Great. So now you're going to open JASP. How to open JASP? Is that the question? Show how to save it again. Okay. One second. All right, so I'll just run through both again. So you're going to go File, Save As. I just like to use Browse because it's the older file sorting system that I like. And then wherever you have your folder for this course, it's going to make sense to have Lab 1, Lab 2 folder. And then in there, first, you're going to save it as an Excel workbook. Then you're going to go to Save As again. Sarah, do you have Excel downloaded on your computer if you're not using the online version?
Yeah, it is on my computer.
Okay. And where are you trying to save it to a local folder on your computer as well, or are you trying to save it to like OneDrive?
Trying to save it to pretty much just desktop. I didn't really have a folder to keep it in.
What happens if you just hit File Save?
Save. Oh, it says we can't save the data, what it's called, because the file is read only.
That's strange.
Yeah.
You do have a tab in the top that says, like a yellow tab that says Enable Editing or anything like that?
No, I already clicked that before.
Yeah, I'm not sure why it's doing that. To all of your, it should be. I'm not sure of a workaround for that beyond downloading the file. When you downloaded the file, where did you save the original?
Just to desktop.
And it won't update that. That's weird. I guess I would try closing out of it and reopening it. And if you lose your data, just let me know and I'll send you a copy of mine or something, or
Okay.
or we can just give you a few minutes to redo those formulas real quick. I can put them back up on the screen. That's probably faster than re-downloading stuff from an email.
Yeah.
Hopefully it saves.
Oh, wait. I think it might have just downloaded. Let me see. Nope, I don't think it did.
So now when you open that file, when you open the original one that you saved to your desktop, it doesn't have all of the work you just did now?
It does actually, so that's good.
Oh, okay, good. So it saved everything. So
Yes.
try again to save it as a CSV file.
Okay. Okay, I think it worked.
Great.
Okay, sorry about that. Thanks for the help.
No, that's fine. Yeah, if that's what I'm here for, that's what lab time is for. If anyone has any questions, we have enough time to get through all of this. So, feel free to ask and we'll try and work through them all. That should be all set. Stop sharing. So now we're going to open JASP, which you may have a link on your desktop, or you may be able to do it just by searching for it. Does anyone have any problems opening JASP? I'll share my screen. So this is basically what you're going to see. And so now we need to import the data that we just created. So I'll go through, if you've been working on something, it will be in your recent files, which is nice and easy. Most of you probably won't have that because we just started this. So if you go to these 3 bars up here, go to open. Under computer, you can go to browse, where you may see your document here. And it's going to show you your Mo data. If all you have is an Excel file, you won't see anything where you saved it. So hopefully this works for everyone. You'll see your CSV. And when you open it, you should get something like this. I'll pause there for a second, make sure that works for everyone. Great, seems like everyone should be there. So now in JASP, the first thing it's going to ask you to do on your worksheet is create a contingency table for 2 variables, bluegrass and plantain. So to do that, we're going to need to know how to navigate JASP a little bit. So a little different than Excel, a little more, a little bit of a nicer layout. Things move a little more organically. You can just drag and drop and pull stuff around. Most of your data is going to be, most of your statistical work is going to be done through descriptives. And this is a pretty nice user-friendly way to look at a breakdown of all your stats. So if we just, for example, take percent soil moisture that we calculated already without you doing anything, just dragging and dropping into variables, percent soil moisture, it's going to give you, mean, standard deviation, and the min and max for that data, which is really fast. A really nice feature of this too is this split. So if we wanted to look at differences in soil moisture against another variable like mowing, you can drag that in a split and it's going to break it down for you. So we've got frequent and reduced mowing regimes, and then soil moisture for each of those. Pretty easy, drag and drop. You can put stuff back. It's pretty safe to just mess around with. So I would encourage all of you to just take different variables, put them in, see what you can do with them, etc. All right, and so we needed to create a contingency table. So we're going to close out of descriptive statistics. We're going to go into frequencies at the top here and select contingency tables. It's going to bring up a really similar plot to what we saw before. just with a bit of a different breakdown because this is going to compare frequency of one thing against frequency of another. You can see on your worksheet, which I guess I'll share that so we're all on the same page. It's asking you to create this contingency table. So you're basically already given what needs to be in rows and columns. So plantain on the top, bluegrass along the side, jasp. So we put plantain in the rows, bluegrass in the columns, and click this little arrow to make it bigger. And then this is going to give you pretty much what was set up on your worksheet, and you should be able to fill in the values based off of that work. So I'm going to close mine out and give everyone just a second to catch up. And is everyone good to go, or does anyone want time to catch up with that and fill out the problem in your worksheet? Okay, so we'll give it 5 minutes and check back in after that, and just unmute and let me know if you have any questions. I'm just gonna grab some water. I'm gonna turn the video off, but I'll be back in. Was anyone able to move on and do the next question as well? The find the mean and standard deviation of percent organic matter for the mowing treatments and put that table in. Oh, okay. Sorry, Isaac. I guess we moved past you a little quick. I thought everyone had the file open in JASP. So were you able to save it to your desktop as a CSV? So do you know exactly, did you save it to a file folder and you know the location of it? It's not going to work if you save it to OneDrive. OneDrive it's a cloud-based folder, so JASP is not going to be able to access that. So you need to save it to a local folder on your computer. I think, did that work for you? Were you able to save it to a local folder? Do you want me to run back through the basics of JASP or are you good? Okay, great. Yeah, we'll just take another minute, make sure everyone's caught up. Um, Great, I'm gonna move on to the next topic then. And we're gonna cover how to make the frequency chart for plant species. So the first thing you're gonna wanna do is going back to your data, which if you were in here, you can just click this far left arrow and that'll bring you back to your data. Or you can X out of whatever you were working on if you don't need it anymore. So then under plant species, any of these titles you can click on and change the type of data. This is what we talked about earlier with different types. So you're going to change number of plant species to ordinal. And then back in descriptives, we're going to take plant species and we need to make a histogram. So we're going to go here. We've got number of plant species selected in variables. And we can go under plots. And here you can make distribution plots or box plots or scatter plots. But these are the two basic ones we've covered today. And it's pretty easy. Once you click distribution plots, it moves it over here for you. It's so much nicer than the output in Excel. Everything's already grayscaled for you. You don't have grid lines or titles. The axis titles are appropriate. So it's just much easier. And then you can, by clicking on distribution plots, copy this. And moving this into a Word document is very similar. You paste. Sorry, that should have been paste as, sure. So again, just with Excel, you're going to want to well, maybe it will let you, if you have the option to paste this picture, then do so. It's not letting me, which I think is probably something to do with how JASP talks with Word. And then it says specifically to format your layout so that it's in line with text. So if you click on your chart, you're going to get this little pop-up here, layout options. Make sure that inline with text is selected. That's just going to be standard procedure pretty much across the course. But it also, it's good to be aware that you can move things in front of or behind text, and that's how you access it. Don't need any of this extra stuff. Could anyone, I think I didn't share the right, did I share the Word document for that? Did anyone miss that? I can cover that again. Yeah, I thought so. Okay, so there, now you should be able to see the Word document. So again, I switched windows without sharing it on Zoom and continued. So it's not going to let you paste this picture from JASP. So you're just going to paste what you copied from JASP. It seems to be putting in this extra info, which you don't need. You can go ahead and just highlight and delete that. And then again, just quickly, if you select the chart, you're going to get this pop up here, layout options, and just make sure it's in line with text. Sure. So someone asked how to change the number of plant species to ordinal data in JASP. You should be able to see my JASP screen again. Going to get out of all of that. So up at the top, where it says plant species, so this column header, if you click here, so it'll say click here to change type of data, and just select ordinal. So just quickly, before we move on, I just want to point out the statistics tab down here. So if you were to put in grass cover and split that by mowing, get this nice descriptive statistics based off of those two. If you break down this statistics chart here, it's just going to give you options to alter this chart. So you can add median, mode, sum, change the distribution, add in some things like variance if you wanted, include quartiles, so like 25th percentile, that kind of thing. And then again, plots, this is how we made the histogram using distribution plot. That's probably not going to look great for the statistics I selected, but that is how you open up all of that. Yeah. Great. So does anyone have any questions? Is everyone caught up? Great. So for your homework for next lab, I can share. You're going to need to create your own data set. So you're going to find at least 10 items around your house. So this could be anything you have access to. It could be, and you need to be able to measure a metric of them. So if you have a kitchen scale, you could weigh things, just better to keep it in grams if you have that option. If you have a ruler, you can measure stuff. You could do length of blades of grass or pine cones or anything like that. You could do pencils in a drawer if you've got a bunch of pencils or pens hanging around. So any data set that you can collect measurements from is acceptable. And you should be able to finish this homework based off of what we covered today in lab. Would anyone find it helpful? I can create some breakout rooms if anyone wants to work on this stuff more together, or I can just stay logged in here to answer questions if anyone wants to do any of that. When is the worksheet due? Someone asked that, I believe. I believe it should be due by next lab. I'm gonna double check with Amanda real quick. So it says on my courses that it's due by the end of today, which I think is in the syllabus as well, that the worksheets for labs are going to be due by the end of the day of that lab. Does anyone feel like they need more time than that? I know there might be some technical difficulties this week. No, we're gonna check in. Someone asked if we've checked in yet. We're gonna do that in a second. I think you should be able to screen share. So we're gonna try and do that if we can, just to show that you filled out the worksheet. Okay, so it seems like people could use more time. So I'm just gonna extend the first one to next week. I'll see if I can do that right now. Great. Sorry if I cut out there. I was changing when the worksheet was due. It seems like some folks could use some more time on it this week. So just for this week's, we're going to extend it to have it due by lab next week. And then in the future, coursework will be due by midnight on the day of labs, which hopefully will have enough time in the labs for you to be able to get all of that done. Great. Does anyone else have any other questions about any of the work? So someone asked if we, Dylan asked if we submit the classwork and homework together. So you're going to show me the classwork hopefully now with a screen share, and that will be your attendance check for the day. And then the homework will be submitted through my courses. So why don't we try and do that screen share now? I'm just going to go in order of who is on my screen. So Stephanie, do you want to can you screen share your worksheet? Hopefully this will work.
I'm trying to share it, but it's not letting me share screen. I don't know. I just got this computer.
Let me check.
It says Zoom will not be able to record the contents of the screen until it is quit.
Oh, okay, yeah. So I need to stop recording. So I'm going to end the recording and